Welcome biologists, and in this session today we're going to be focusing on multiple alleles. So in the previous two sessions we've had a look at monogenic inheritance and dihybrid inheritance, and today we're going to be focusing on multiple alleles. Now, as with the other examples there, we have to make sure that we're using all the examples and using all the 4 points within this little box here to pick up the marking points. So I'm going to introduce you the main example you get with multiple alleles, and then we'll go through some examples of how to do those crosses. So with multiple alleles, you normally get questions to do with the blood groups and blood types. So as you can see here, in order to get a blood group A, I can have different versions of the same gene. So I can have multiple alleles that code for my blood group A. I can also have AB. I can have B, so for blood group B, I can again get multiple alleles there that code for B. And then for group O, I have 2 of the IOs. So here I've got multiple alleles. I've got IA, IO, and AB. And different combinations of these alleles can produce different phenotypes. So underneath there, you've got 3 questions to do 3 genetic crosses. I'd like you to pause the video and have a go at doing those crosses based upon the genotypes there in the table. Now, don't forget to pick up all your marks. You need to do all 4 things within this box and each question to get all your marking points. So please pause the video now and have a go at doing those crosses, because I'm just about to now go through those answers. So in the first question, it asks us, it's very generous here, and it gives us the genotype of the parents. The next thing that I need to figure out are the possible gametes that could be produced from these genotypes. So there they are, and they're in circles to show the examiner that I know these are the gametes produced. I then put those gametes into the Punnett square and complete the cross. Now, the last thing I need to do to pick up my marks here is to generate a genotypic or phenotypic ratio, depending upon what the question asks you. Here we've got both, so you can see both here. So the genotypic ratio here at the top, and then the phenotypic ratio here underneath. But you have to make sure you either put one of the genotype or the phenotype. You can't just state normally. You don't just get marks for stating 1 to 1 to 1 to 1. You have to say 1 to 1 to 1 to 1, and then AB to A to B to O. So you have to be a little bit more specific with your answers. The next question was asking for a father that has a blood group of AB and a mother that has a blood group of O. So the genotypes for these parents are as so. I then need to draw out the gametes that are possible from these genotypes, which are listed there. I then put them into my cross, into my Punnett square, and complete the cross. And then last of all, I need to do the ratio. So I've got 1 to 1, A to B for this particular example. And the last question, so homozygous is where I have the same version of a gene, and heterozygous is where I've got different versions of that same gene. So for example, here I've got the same version of the gene, so IA and IA for the mother, and then for the father who is heterozygous, I've got different versions, so IB and IO. And they need to work out the gametes present that can be derived from those gametes. I put them into the Punnett square to do the cross, and I then work out my ratio. So there we have it. That's an example of multiple alleles where I have more than 2 alleles that can help to code towards a phenotype. As ever with these questions, please do not forget that you need to include these 4 main things here to pick up all the marking points. Please read your questions carefully and annotate them to pick up as many marks as you possibly can. And guys, good luck with your exams.